Hi, dear students. Welcome back to one more session of molecular biology. And today we're going to talk about the translation which happens in prokaryotes. The term translation is quite familiar to all of us because it is a process of protein synthesis. And this protein synthesis normally happens in the case of prokaryotes as well as in eukaryotes in certain organelles called the ribosomes. And in the case of prokaryotes, we can see that these ribosomes are being found within the cytoplasm, and we do not have a clear-cut distinction between a nucleus or nucleus and a cytoplasm because the nuclear membrane is totally absent in the case of prokaryotes. So when you go to consider translation in the case of prokaryotes, the process of translation is being closely linked with transcription also. As we know, translation is a process which is following transcription. That is, DNA is converted into RNA, and that RNA is further converted into protein in the process of protein synthesis or translation. So, in the case of prokaryotes, since there is no much demarcation between the nuclear membrane, nucleus, and the cytoplasm, the process of transcription and translation occurs with in a high frequency with not much time of gap or anything like that. So today now this is a seminar which has been held by Krishna Priya, and she'll be talking on this, and later on I will be giving my feedback on her presentation.
Hi everyone, myself Krishnapriya. I am taking about the translation in prokaryotes. I will explain about the translation which is happening in prokaryotes in two videos. So after watching my first video, please go to my second video too. So what is translation? Here you can see a pictorial representation of translation which is happening in the prokaryotes. Translation is simply the protein synthesis. It involves commonly linking amino acids together to form a polypeptide. It is decided by mRNA and is conducted in ribosome. The direction is from 5' to 3'. The main features: translation begins when a ribosome binds mRNA and is positioned properly so that translation will yield the correct amino acid sequence in the polypeptide chain. tRNA molecule carry the tRNA molecule carry the amino acids to the ribosome so that they can be added to the polypeptide chain as the ribosome moves down the mRNA molecule. Polypeptide synthesis begins with an amino acid at the end of the chain with a free amino group and moves in the C-terminal direction. Thus, translation is said to occur in the amino terminus to the carboxyl terminus direction. Ribosome in prokaryotes. Sedimentation value is 70S with 30S and 50S subunit. They are free in the cytoplasm if protein is not being synthesized, and they come to form ribosome only when translation occurs. The main regions are translation domain and the exit domain. The translation domain is the region where the translation occurs, and the exit domain is where the growing peptide chains emerge from the large subunit. Here you can see the translation domain and the exit domain. The picture is taken from the Prescott book. Each subunit of the ribosome is made up of extraordinarily complex structure constructed from one or two RNA molecules in many polypeptides. So what are the role of these RNA molecules? It contributes in the ribosome structure, helps in the initiation, and also has the catalytic role in protein synthesis. The cysteine RNA molecule of the 30S subunit is needed for the initiation of protein synthesis. The 3' end of the cysteine RNA complexes with a site on the mRNA called Sine-Dalgaro sequence, which is located in the ribosome binding site. The cysteine RNA also binds a protein needed to the initiation factor 3, and the 3'CCA end of the amino acid tRNA. The 23S RNA has a catalytic role in protein synthesis. Polyribosome or polysome. It is a complex of mRNA with several ribosomes is called the polysome or polyribosome. In prokaryotes, coupled transcription and translation occurs. This is a pictorial representation where you can see the transcription and translation occurs simultaneously. Transfer RNA and amino acid activation. The first stage of the protein synthesis is amino acid activation process, in which amino acids are attached to the tRNA molecule. The receptor or amino acid stem holds the activated amino acids in the 3' end of the tRNA. The 3' end of all tRNA has the same CCA sequence, and the amino acid is attached to the terminal adenylate acid. At the other end of the clover leaf is the anticodon arm, which contains the anticodon triplet complementary to the mRNA codon triplet. There are D or DHU arm, which has the unusual pyrimidine nucleotide dihydrouridine, and the T or T5C arm has ribothymidine or pseudouridine. It has also a variable arm. Here you can see the variable arm, D arm, T5C arm, acceptor arm, and the endocodon loop. Amino acids are activated for protein synthesis through a reaction catalyzed by amino acid tRNA synthetase. Amino acid plus tRNA plus ATP in the presence of magnesium 2 plus gives the amino acid tRNA plus AMP. Here the ATP is converted to AMP, not the ADP. Amino acid is attached to the 3' hydroxyl of the terminal adenylate acid on the tRNA by a high energy bond and is readily transferred to the end of the growing peptide chain. That is why it is called the amino acid is said to be activated. Amino acid tRNA has the same role of the DNA polymerase, which has the quality of the proofreading. If wrong amino acid is attached to the tRNA, it will proofread. Protein synthesis. The stage of the protein synthesis is divided into 3 stages: initiation, elongation, and termination. Initiation. Prokaryotes begins protein synthesis with a specially modified amino acid tRNA and formyl methionine tRNA fMeth. When a methionine is to be added in a growing peptide chain, a normal methionine tRNA meth is employed. Then next, the 30S subunit possesses a molecule of cysteine RNA with nucleotide sequence that are complementary to the signed Delgaro sequence in the leader sequence of the mRNA. This is because the role of the leader sequence is to align the mRNA with the complementary bases on the cysteine RNA of the 30S subunit, such that the codon for the initiated fMeth tRNA is translated first. Initiation codon AUG or GUG binds to the fMeth tRNA anticodon. Finally, the 15S subunit binds to the 30S subunit mRNA, forming an active ribosome mRNA complex. The fMeth tRNA must be positioned at the peptidyl or P site. You all know that there are 3 binding sites, P, A, and E. Here the fMeth tRNA must be positioned in the P site. Initiating factor 3, IF3, prevents the 30S subunit binding to the 50S subunit. IF2 binds to the GTP and the fMeth tRNA and directs the attachment of the fMeth tRNA to the P site of the 30S subunit. The GTP is hydrolyzed during the association of the 50S and 30S subunit. The third initiation factor, IF1, appears to be needed for the release of IF2 and GDP from the completed 70S ribosome. IF1 may also help in the binding of the 50S subunit with the 30S subunit. It also blocks the tRNA binding to the site, to the A site. The next 2 stage, elongation and termination, I will explain in the second video. So after referring to my first video, please go to the second video too for clear understanding about the translation which is happening in the prokaryotes. Thank you.
Hi dear students, so let me just give a consolidated brief report on what is translation in prokaryotes, which was being given on the first video put forward by Krishna Priya. We can see that translation in prokaryotes mainly happens in the 70S ribosome, which consists of 2 different subunits. One is a 30S subunit, which is a small subunit. It has been formed at the bottom. And on top of this 30S subunit, there is a bigger subunit here that is mainly considered to be as a 50S subunit. And associated with the 30S subunit, there is a particular ribosomal RNA called the 16S ribosomal RNA. And associated with the 50S subunit, we have another RNA called the 23S ribosomal RNA. These 2 RNAs have 2 different functions. Now, 16S ribosomal RNA, it mainly interacts with the mRNA, which codes for the particular amino acid sequence, or the codon has been present on the mRNA. So the role of the 16S rRNA in the 30S ribosome is to interact with the mRNA. And on the 50S subunit, we have the 23S ribosomal RNA, and it has mainly what is it? The activity, that is the peptide bond formation, is being carried out by this 23S ribosomal RNA. So you have a ribosome which is having two different subunits, and in both these subunits they have two different roles. Now, the step of protein synthesis in prokaryotes as well as in eukaryotes, commonly speaking, it consists of three different steps. One is the initiation, the second one is the elongation, which happens after the peptide bond formation, and ultimately you have a termination of the protein synthesis. So these three steps are all happening within the ribosome itself. So how does this happen? So within the within the ribosome itself, there are three seats which are possible. That is at three different places. If you sit in one place, one step will happen. If you sit at another seat, you have another function. Okay. So what are these steps? I'll just show you a picture of that right now. So now if you look into the ribosome, you can see that it is, as I mentioned earlier, it has got three seats here. So in three seats, the first one, now in which seat will the first amino acid comes and bind? So that's what I came and put over here, the podium. We always want to become the first, and the first is always being present in the middle. So what happens, the first amino acid which comes during the protein synthesis will always occupies the middle seat, and that is called the P seat, or the peptidyl, or the donor site. Okay, and the second one here, or the E seat here, is the exit seat, and the one which is being found towards the right of the B seat is referred to as it is referred to as the A seat, or the amino acyl, or the acceptor A site. So for the initiation of protein synthesis, what happens? The amino acyl tRNA has to be formed, and that amino acyl tRNA, especially the formal methionine bearing tRNA, will have to come and bind towards the P site. And always, once it is occupied by the P site, just the second amino acid will be put towards in the A site. P doesn't want anyone to sit close by at this side because it has to go towards this direction. So the second amino acid will be always being present here. Now why it is being present here, you can just use your common sense, because the mRNA is being present here. The start codon starts from here, and it's towards in this direction that the amino acids are being read. So the second amino acid will be kept in the A site. So do not get confused. The first amino acid which is coming will come into the P site, and the second amino acid will come in the A site. Now regarding the initiation of protein synthesis, just imagine that 30S subunit is linked with I initiation factor 3. Remember, 30 linked with 3. So IF3, they are being linked together, they are floating around the cytoplasm and they are walking. And then the cell needs that protein synthesis has to happen. So what that, along with that, that interacts with another molecule referred to as, so when this is moving, at the same time you can also find that formal methionine is also roaming around in the cytoplasm. So formal methionine has to be brought towards the 30S subunit. That role is being done by another mediator called the IF2, which binds with GTP and goes and attaches to the formal methionine. And it will drag and bring the formal methionine and put it in the which site? Do remember, in the P site, in the P site of the 30S ribosome. Now, at the same time, of course, that is possible only if the mRNA is also being bound. So, the 16S ribosome will interact with the, here in the 30S subunit, you have the 16S ribosome. The 16S ribosome of the 30S subunit will interact with the mRNA, and that interaction will be possible over here. And we also have the formal methionine which will come and bind to the P site of the ribosome, of the 30S ribosome. Now, in the next step, what happens? The 50S subunit has to come and bind. And you can see that once a phenylmethionine has been bound here, the initiation factor 1 does not have any role, and so it has to go. So for that, we have a third initiation factor called the initiation factor 1. Okay, and initiation factor 1 will help to remove the what initiation factor 2, and at the same time what happens when the 50S subunit comes and binds over here, it will help in the binding of this 50S subunit to the 30S subunit. And further on you can see that for GTP has been broken down and GDP has been released, and this will cause the formation of a complete 70S initiation complex where the phenylmethionine bound with the tRNA is being located at the P site of the ribosome, and the E site is left vacant and the A site is being found vacant, and you can found that the tRNA is being interacting, the anticodon of the tRNA is interacting with the what? The mRNA, which has been found to the 30S subunit of the 70S initiation complex. So the first role of initiation factor 3 was, it was a one which was keeping the 30S subunit from binding from the 50S subunit, and once the initiation factor 3 was removed, the chances of binding of the 30S subunit with the 50S subunit increases. So this is what we have to talk about the initiation of protein synthesis. We'll be talking about the next steps in the coming videos. Thank you for now.